Hi, I'm Susan and this is Hayley, and welcome to another episode of Cancer Research Demystified. Today we're going to talk all about tissue.
So tissue can be a number of different things. It could be that whole organ you get taken out during surgery, but it could also be a smaller sample of skin. It could be a biopsy you've had. It could even be saliva. So it's just a collection of cells.
Right, so first things first, there's probably a reason why your doctor has decided to take this tissue from you. So it's going to go straight to the hospital lab, where doctors called pathologists will have a look at it and decide what needs to be done next, whether you need to be given some more tests or treatment or so forth. But after that, if you've agreed to take part in research, that's where we come in and we get to take a little sample of the tissue that's left over.
So in this video, we're going to go into what happens to the tissue once it's left your body. So how long does it last? How do we make it last longer? Where do we store it? How do we process it so we can carry out cancer research on it?
So to show you an example of how we would process tissue that comes into the lab from a biopsy or surgery, we're going to use an example of a mandarin today, a little less painful than taking some of my own tissue. So this is a biopsy punch. It can be used to take a skin biopsy, but today it's going to be used on our little orange here. So once we get the tissue back to the lab, we can store it in a number of different ways. Most usually we'd freeze it really cold for molecular analysis, or we can store it in these wax blocks. These can actually be kept at room temperature, and then when we need them, we can take really thin slices of them, put them on a slide and look at them down the microscope. So if we're going to freeze the tissue, then in order to preserve all the molecules inside, we need to make sure that we freeze it as cold as possible, as quickly as possible. And to do that, we're going to use liquid nitrogen, which is why I'm dressed in this rather fashionable getup. Liquid nitrogen is minus 196 degrees Celsius, which is twice as cold as our freezer, which is pretty much as cold as the coldest temperature ever recorded on Earth. So we're talking pretty cold here. To freeze it, we put our little piece of mandarin tissue here in a tube and drop the tube inside the liquid nitrogen, where we hear this nice fizzle, and then it's all ready to go in the freezer in a few minutes.
So this isn't quite so cool as what Susan's just shown you. And what I'm going to explain is how we get things into this wax block. The first step, you just put something in some liquid. But what's special about this liquid is it's called formalin, and what we're doing is fixing the sample. So say you go to the Natural History Museum and you've seen all those animals, all those samples in like yellowing liquid in jars up on shelves. They're all fixed, they're preserved, and the liquid is this formalin. So what this liquid does is it kind of makes everything in your body stick together. So it bonds them together. All the different molecules will just suddenly stick to each other. So everything is stuck to everything else. It's not going to decompose, it's not going to react, nothing's going to change, and the whole sample's preserved. So we have little colourful cassettes. They have a dual purpose, a stand for our block, as well as a little cage to stop our samples escaping. And then they get fixed in formalin. So we then leave it in this solution for a couple of hours, depending on how big the tissue is. Once we've fixed it, the next step to turn it into this wax block is to remove all the water molecules and replace them with wax molecules. First step, we use a machine called a tissue processor. Inside, we've got a load of different bottles and a load of different solvents. And they're all different gradients, all different concentrations. And the idea is because obviously water and wax don't dissolve in each other. You know, your candle doesn't melt in the rain. So what we're trying to do is we need a solvent that dissolves both water and wax. So we have our sample with the water molecules. All of that goes into this solvent. The solvent then replaces all the water, and then the wax comes in and replaces all that solvent. And then we end up with everything in hot liquid wax. So we just pop this in here. And we run it overnight. So after the processor, we take it to a machine called the embedder. And what we do with our sample is we just drop it into hot wax. And this is just while we wait and set up the machine. We definitely do not play with this in the lab and make our fingers into candles. So all we basically do here is we're taking it out of its cage, we're putting it into a mould, which makes our wax block. We then fill this with wax, put its little hat on, which holds the block, and put it on a cold plate to cool.
So now this tissue has become rock hard and it's set in this wax block, and we're ready to take a really thin slice off of this so that we can then put that on a slide and look at it down the microscope.
So Susan's put the block in this machine here. We call this a microtome. And what she's so expertly doing here is she's slicing it super thin, so we can get thinner than a single cell, and then we can see through it, we can get light through it and see it on a microscope.
So this machine holds the block securely and moves it up and down over this very sharp blade. So once we have this thin slice, we put it into a water bath, and this just makes it easier to get it onto a microscope slide. As Hayley said, we're cutting a 5 micrometer slice, and if you picture the smallest divide on your ruler is 1 millimeter, 1 micrometer would be 1,000th of that. So this is 5,000ths of one of those small divides on your ruler. So rather than looking at this slide down the microscope straight away, we tend to add a number of different types of dyes and stains, and these help us to see in different colours the different types of cells and the different structures within the tissue. So the old-fashioned way is to add drips of dyes straight onto the slides ourselves, but nowadays we've got robots that do that for us. So we put these slides in here, press go, tell it what stains we want added, and then go off for a cup of tea. So after all that work over the course of a few days, we've finally got our beautiful slide stained with human tissue, or in this case mandarin, and we're ready to look at it down the microscope. So here are some examples of some tissue that we've stained in the lab. You can see the different types of cells that make up the tissue and how intricately they're arranged.
So each purple dot here is the centre of a cell, and you can see there are hundreds of cells within this tissue, and you won't be able to see each of those with the naked eye alone.
The different stains that we use help us to see how quickly cancer cells are growing, or which area of that tissue is cancer and which is healthy, that kind of thing.
So the most recent research is trying to get the staining to be really specific. So we want to look at not just patterns and shapes and general stains throughout. We want to look at one particular molecule maybe that we're interested in, a particular protein, a particular sugar, all sorts of things that are found in the cell. Because we're starting to realise that some of these particular proteins can tell you things about the cancer. They can tell you how maybe it's formed. They can tell you how aggressive it is. They could tell you if it's going to spread or not.
And as we learn more about these particular molecules, usually proteins, it can help us to develop tests for the future. So we might be able to do a tissue test on biopsy or surgical samples in the future that can tell us more about that patient's cancer. For example, how aggressive it is, or maybe even what type of treatment that patient should be given next. So that's all for now. If you have any questions about tissue, how we collect it, process it, and why, then please put them below in the comments. And other than that, we will see you next time. Bye!